moving into round 2, or exam 2 material. Yeah, so next class we'll spend a little bit of time on exam 1 to go over it. You can kind of ask any questions, and then I'll table it for office hours or after class. So I like deal with the most commonly missed, maybe open it up to one more question if somebody wanted to ask something. But that's kind of where that's at. I'm just going to remind you, though, if you didn't do as well as you wanted to on exam, it's definitely a category of some of you guys that I want to see you guys come get better. It's really about time spent with the material. So this lecture today, if you don't revisit it till about like the week before the exam or a couple days before, you're going to be relearning it all, right? Instead of revisiting this within 24 hours, review it, all the way up to the exam, and then this stuff you have hardwired. All the stuff that you're going to be learning is just the last couple lectures. If you do that, you do yourself a service, because all this stuff will be like old hat, right? Instead of new hat. Because by the time you get to the exam, and I know there's a lot of people in this room like me that did it that way, cram it all at the end, and then you forget all the material that you just got delivered, because it's going to be several weeks away, right? Just saying, if you're not getting what you think you wanted out of your performance, you have to consider another option. What else are you going to do different? If you do the same, you might get the same result. All right, and I'm always willing to help you. Come to my office hours, and I can help talk about study habits and any of that stuff too, and go over material, of course. All right, anything from you guys? Nothing? Good to go? All right. Oh, by the way, you're always welcome to hit up recorders up here too. So I just thought I said it probably the first day, but you can always bring up a recorder. You can't videotape me, but you can record, audio record. Yeah.
Do you happen to know, like right offhand, like how many points was added to everyone's test exactly?
How much was added?
Yeah, like how many points?
I'm gonna let that just be a Tuesday thing.
Okay.
Yeah. But I'll say this, there was a perfect score. So it was doable. All right, okay, so kind of right here is the nuts and bolts of genetics that takes a turn from Bio 2 material that you've heard a lot of. You should have heard replication, transcription, and translation. And then we added stuff to it, right? This stuff you probably haven't heard. And then this whole section, exam 2, I'm going to say it's some of the harder material because you haven't heard it before and you're learning some new concepts right here. Doesn't make it, to me, really harder per se. It's just that you just got it. It's newer stuff to you. Exam 3 is going to be Mendel, right? You've heard Mendel since probably middle school, right? And then linked genes will be your tough topic in that one because we kind of switch a paradigm when they're found on the same chromosome. But in this one, gene mutation is fairly straightforward, I think. You just got to put the time into studying it. And then the harder stuff is prokaryotic regulation. That's the one that comes up after. And the lac operon is kind of a little bit challenging. How does the regulation steps work, right? So I'll make you known where I kind of come up where it's kind of, you're going to have to spend a little more material and it's a little bit of troubleshooting or thinking through problems. All right, so we're going to start off with, so this is obviously not right? You got 3 sets of eyes. Probably is more like a developmental process that went awry of setting up 3 optical sort of things. So, but bottom line, this is the question I want you to think about. And I'm going to hit an eye clicker on this one. All right. It's kind of an opinion, but it's kind of it's here to make a point. All right, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1, and let's see how many people are mutants out there. All right, you're all mutants, except for 3 of you. 3 of you, I guess, is the prototypical perfect individual, right? Which doesn't exist, right? So we are an accumulation of all of our mutations we've been getting through our ancestry and continuing. So we are mutants. We are adapting to our spaces, often through mutations. Some of the mutations. This is what's interesting about this kind of a question. It gets into the idea, is mutant good or bad, right? Well, if you're into superhero shows, yeah, right? It's not bad to have some of those skills, right? There's some other superhero shows where they actually go opposite direction. I forget that one on Amazon. But ultimately, we think of it as a bad thing, right? But truthfully, that is how we also adapt to our circumstances and how things kind of evolve and change. Now, typically, if you have like an engine that's running perfectly and you start tinkering with it, mutating or changing it, the engine of like, finely tuned Porsche or something, probably is not going to go well, right? So if we're like tuned to our environment or enzymes, you start tweaking things, often things can go bad. So that's kind of why we're based on that idea that a mutation doesn't really sound good, right? But it could be. It actually could be. But it also could be, so what? It could be like, I have a mutation and it really didn't do anything. So you got 3 categories. Some that are good for you, some that are going to be bad for you, and some are like, whatever. Didn't really do anything. So we are all mutants in the essence of it. So here's a key definition. Right here. See how it's underlined? Really important detail. And for some of you guys who are having a tough time decoding where the test is coming from, I pretty much give you all the answers up front. That's going to be something that's going to probably be on the test because it's an important detail, right? So I'm typically going after the really important concepts. Now, the reason why this is an important concept because a nucleotide could change. That is the first idea of a mutation. But then if it changes back to its original condition because of a repair, if you repair it before the damage is really done, when is the damage done? When the cell divides and hands off the mutation to descendants is the moment that you call heritable. So that calls a trajectory of all the cells that have had that change in their DNA sequence continue to propagate it on and on and on and on. Now, you eventually might mutate it back, but already you've got a whole bunch of cells that are mutated, right? So we can talk about second mutations later, and we will. But the first primary mutation has to be passed on. And if you can repair it before the damage is seen, or it might not even be damaging, it's just a change, right? And then you change it back. It's like a tree fell down or something that you never saw. Like, you never know what tree ever did, went down. Maybe a tree went up in its space. You never figured it out, right? Point is, we have to detect it through heritability, right? So it's going to follow that line. Now, this is what I already said. You got the positive, you got the negative, and then you got things that don't have any noticeable effect. So I already kind of covered that concept. And so we are going to follow through on these kind of types of mutations, but those are when they're under some kind of selectional pressure when they're here. What do I mean? So natural selection and evolution is involved in like, is that good or is that bad? There's a change, and then you decide what's going to fall out from there. And if there's a selectional pressure, and it says good, bad, or it didn't really do anything for me, right? Didn't do anything. So, and there's the repair part. That's the end part of this lecture that we're covered where, remember, there's a lot of DNA polymerases that aren't the ones involved in replication. Even in E. coli, we had 5 DNA polymerases, and we only talked about 1 and 3, right? So you got 2, 4, and 5, and they all fit this category. So you have 3 versus 2, and then eukaryotic, you have more. You have way more repair type. All right, so this is where we're going to go with this. Single gene mutations is where we start. Those are what we call point mutations, where they happen at a very specific space, and it's local, and it's happening right there in this small little area. You can still have insertions and deletions. We'll cover that later. But when you get into chromosome mutations, you can have a deletion of a chunk of the chromosome. And if you do that, whole chromosome lopping off a chunk of it, they're talking about like tens to hundreds, possibly thousands of genes. Right? So we're not talking about small time, we're talking big time we move here. And so then this will cover other things you probably heard. You got chromosomal gain, loss, duplications as the gain, deletions for loss, and then you have translocation and inversions. So those are the 4 biggies that we'll cover later, also in lab. And then we move to genome mutations, which is changes in chromosome number. And that would be like your trisomies, like your trisomy 21. and versions of that. If you were in my lab today, you heard me mention Turner syndrome, XO. Again, that's another version of a genome type mutation. You gain or lose chromosomal material, the whole chromosome. Or polyploidy means you can go from 2N to like tetraploid 4N. Octaploidy, you can get up to quite a few numbers of your chromosome. Chromosome 1 would be 4 times rather than diploid. So that happens in plants more frequently, but we have amphibians and stuff like that happens too. All right, and that's where we'll finish the exam. We'll kind of be finishing around here, but sandwiched in between here, we also have regulation, prokaryotic and eukaryotic. All right, that's kind of an overview. Let's get into the nuts and bolts of mutation. All right, so it's a hierarchy kind of philosophy. So you're going to start with just classifying the mutation at hand. So the first level is what changes. So actually, let's go to the definition. Point mutation means at that place. At that very particular location, something's going to happen. Now, it could be a change in a base, and that would be called like a base substitution. You're going to swap it out for another one. Or you can have an insertion or deletion, in which the classic way that that's often discussed is like, let's do it with black. That's how they're often referenced, even though it's kind of jargony. It's actually pretty well ingrained into the vocabulary. In, del, insertion, deletion, indels. So often that's the term that gets kicked around. Now that's going to offset things, right? So when you insert or delete, those were often the consequences reading frame changes. Remember we talked about the reading frame is set up by the ribosome, and that you read everything in 3s. Well, if you offset the registry by 1, then everything's read differently, right? That's a big consequence. But if it happens in a non-coding area, let's just say an insertion deletion happens in a repeat, STR, for example, it's no consequence, right? It's like, so what? There's no ribosome reading any of that, right? So again, the connotation of is that good or bad comes with selectional pressure, which means it has to have some expression, because just messing up an STR repeat's not going to do anything, right? All right, so the way I think about this, all right, it's a little bit of sports. Sorry, throw it in every once in a while. So if you have a substitution in a game, you have the same players out there. We'll go with the Kings basketball team. We got 5 players out there. And you're like, all right, go ahead and take the seat while you swap somebody else, right? That's a substitution. When you talk about an insertion or deletion, when we get to that, the analogy will be like a penalty, in which, let's say in hockey, they actually remove a player for a full-on penalty, and they actually have to take a seat, and now you're playing down a player, right? Or in soccer, you get a red card, and you lose a player, right? You actually have to play with one less. So that would be you're down a member, or you get an extra one on the field, right? So think about, that's what pretty big game change right there. But swapping one now comes down to 2 different definitions, and it's got to be on the test. So when I say that, red flag it, right? So a transition is when it's of the same category or class, and you do still, remember the old vocabulary, purine and pyrimidine comes right back. So that's why we can't lose the last material. We're going to keep building on it. So a purine to a purine, or a pyrimidine to a pyrimidine means the same class of type, a single ring for a single ring, or a double ring for a double ring. All right, so having said that, what we're going to diagram over here is so it's a little bit of a matrix right here. All right, so this would be, all right, so if I circle, the mutation is going to be this one. Just take that left corner. So the A can turn to a G, and that would be a purine, right? So that would be, it has one option to do that, to turn into the other double ring type. But if it goes this way, or this way, those are your transversions. So transversion means change to the opposite class. Okay, so A turns its double ring, turns a single ring, or vice versa. Now, this statement down here is the conundrum that we're going to deal with. Because when I'm looking at this, that A has one possible purine change, and yet two pyrimidines. So the way we're going to discuss this is we're going to have transversions. Okay, so now this is going to be the category that's possible. What is possible to happen? Or potential. I'll write that underneath it. So I'm going to call it is exactly this, like what in nature could happen. I could get one transition and yet two transversions right here. Everyone follow me? That's straight up what we see as possible. Now, what we actually observe in nature or what exists, what do we, when we actually look at organisms and we compare them, you do a sequence comparison, and you look at what changes and how many times does it happen this way, you see the exact opposite. So transitions are more likely to happen twofold more than a transversion. You go, well, the math doesn't work out on that. It's actually reverse. So the conundrum goes like this. The reason how you can solve it is that in nature, it's favored more to have a transition type because it's a gentle type of mutation. So if natural selection is going to act on something, when you have a radical change, it's a little bit more abrupt, right? So transit, now I want you to think about the DNA itself, just fundamentally, you have like a two-ring alternating with a one ring, right? So you get, they can swap sides, two ring, one ring, but it, all up and down, it's got to be evened out, that there's always a 2 to 1, a 2 and a 1 of the rings. That's what I'm referring to here. And so that makes everything nice and easy, like a ladder that would be systemic. Now, of course, the ladder is going to twist, but it's still, if I turned it back, it would still be nice and even and regimented across there, right? Now, imagine if I put 2 of these in. You would pinch in, right? You would basically have like a little hourglass. I'll draw it like that. So one ring and one ring would make it go in. And how about the other way? It's going to bulge outwards. And that would be, so that's not consistent, that's not consistent, this is nice and even keel. So repair enzymes would recognize that as an abnormality more frequently than just like so imagine if this is a G and you switch it to an A. That's a transition. And that transition would keep the same distance. And if you're a repair enzyme, you're like, everything looks good over here. If you're just kind of like just feeling by braille, like, yeah, it just seems fine. Everything's nice and uniform. But then I feel this, I'm like, what the heck is that? Bulge or pinch. You're going to repair that more frequently. So that's one explanation. The other explanation is hardwired into the genetic code. If you remember the genetic code, when you actually have a purine going to another purine, it's more typical that that stays the same amino acid. Remember the degeneracy? So it means if you mutate it, you still get the same amino acid out of it, commonly, not always. But basically that'd be like, yeah, so what? That's the so what, right? Because it's going to be called in a couple minutes silent mutation. Because you mutate it, there's no consequence for the amino acid. And it's like, yeah, whatever, we can absorb that, right? So transitions turn out to be softer, and they actually go a little less detected, and you do a transversion that's more hard jolt to the system, all right? So that's helping explain that ratio there. And I often test on that, okay? Things I go over on the board with a little more detail tend to end up on exams. All right, so basically now we've just categorized baseline. What kind of a change do we have? Now we're going to go into some of the consequences. That means, okay, first level is, if it happens in an STR, who cares? We're done discussing it. It's neutral. It means it's basically no change to any protein. There's no change to an RNA. You're like, whatever, it's going to be fine. You change some junk DNA or something, whatever. But, so that's going to happen. And actually, guess what? How many times is a DART mutation going to hit an exon? Does anybody remember the percentage of the genome that's exons? 2%? Perfect, 2%. 98% of the time you're hitting something else. Right? So, okay, just focus on that for a second and let that seep in. A lot of mutations can happen without consequence. Now, you do hit an exon. So now let's talk about the next level of hierarchy on, here's the normal sequence, and here's where it changes at this position. So relative, it should be an A, it is now changed to a G. So what we're going to look at now is the amino acids that are derived at X. It's an exon. And we say, oh, that turns out in the third position. What's the third position called, you guys? Wobble. The wobble, because any of the four are going to remain that same leucine. So you can absorb that change, and it's no consequent. And silent means you will never notice it, because selection is going to be acting on the protein, not on the DNA. Right? It's acting on the protein. It's the same exact protein. Now, I'm going to say another word in a couple minutes called neutral. Neutral is not the same thing as silent, and I will be testing on that. The reason why I'm harping on that is neutral is a missense mutation, a type, that's going to change an amino acid. And when it changes the amino acid, okay, so let's actually do that first before I talk about the consequences. That's T is now going to become a C. So it becomes TCA. When you go to the genetic code and look it up, you're like, oh, that's serine. It's not leucine anymore. Now, the amino acids have changed. So first of all, let's talk about the word. I know it's kind of bizarre, but take the word miss out of it and just say the word sense. When we talked about codons, we talked about in two classes, sense versus nonsense. Does anyone remember what nonsense referred to? Stop codons. It doesn't mean anything in the coding system. It means just stop, right? There's no amino acid coding for that. And sense means it codes for an amino acid. Thus, the next term, oh, if it's a nonsense mutation, it has something to do with stop codons. So it's an archaic, I know, verbiage or vocabulary. It's based off of the original sort of kind of concepts that were generated. I know nobody's really flipped names. But the idea is that, let's go back to missense. It still makes sense, right? But it's missed the correct sense of what we originally intended. So we missense. It's an amino acid change. So let's actually say it clearly. It's a nucleotide substitution, point mutation, that changes the meaning of the amino acid, or it changes the amino acid. So now you're swapping out an amino acid. Now you have to ask yourself, is that meaningful? And there's where selection comes in. So first of all, that serine may be really detrimental, cause possibly lethal, right? You know, hobbling is also, that's not good either. So if you're, totally maimed or death is totally bad stuff. But the deal is it could be neutral. It could be like, you know what? This isn't even in the binding pocket of what it's doing. The enzyme has a very important shape, let's say. You've now altered its structure, by the way. You have altered it. But is it meaningful? And if it's in the active site of where the enzyme does its business, that's going to be bad. Or it could actually be good. So we could go the opposite way, bad or good. But if it's in a location, it's like in its tail, and it's like flavoring, I call it, like more just, what is the shape of it, but the business end of what it's doing, if you think about a Pac-Man, is doing all the enzyme crunching, but on the backside, you put a little divot in there. And it's like, I don't care, I can still crunch, right? So it really depends, so it's very context driven, right? We're going to talk about sickle cell anemia a couple times through the course, and pretty shortly we're going to talk about nobody really wants to have sickle cell anemia. Not a good thing, right? Because anemia says you're losing blood, you're anemic. And it's only one mutation that generates the sickle cell condition. One mutation that leads to one amino acid change and compounds into the problem. So one mutation can do it. But it also could be fine. Now, typically, I'm not going to test on this concept, but I want you to kind of think about this a little bit. Well, actually, it is testable in this kind of way. Because neutral is this idea that if you basically take a hydrophobic amino acid or nonpolar, and you change it to another hydrophobic. Is that possible? Of course. There's a whole bunch of hydrophobic amino acids. Okay, it could be alanine and valine. Alanine and valine are hydrophobic. So if you swap it between those two, do you think there's going to be a lot of radical change in the protein? No. How about if I swap out a negative charged amino acid, aspartic acid, and then you change it out for another negatively charged amino acid, the same class? It's charged, right? It's going to do the same kind of structurally. So that's a soft type mutation that if you have the same class and similar type of amino acids that you change, then you often have neutrality. You change it radically to something that's hydrophilic on the surface. This is going to be the sickle cell anemia story. And you change it to a hydrophobic one, and it's still on the surface. You think that's going to be good? No, and that's what creates the sickle cell effect. You change it from one class to another, and it's like, oh, I don't like being on the surface. I'm a hydrophobic amino acid. I want to be buried inside of the, hiding away from water, not exposed to it. All right, so that's what I wanted to get you to see. Structure leads to function, right? We say that all the time, but that's kind of what it comes down to. Right there, the structure is changed. And actually, serine is quite different than leucine. Leucine is hydrophobic, and serine is polar. So that probably would be bad juju right there. All right, so anything on missets?
If that did happen to somebody,
Yep.
like it went from like hydrophobic to polar,
Right, amino acid.
yeah, what would happen?
So again, it's still context driven. If it's really in a place that's non-consequential to the function of the protein, because there's only certain places on the business end, then it might still be fine, right? So the question is how important the protein is, right? So if you have, okay, how about coloration? Albino. So can albino things live?
Yeah.
Sure, they live fine life, right? In fact, on a pet trade, there's a lot of money involved in albino snakes and all that stuff, and and the bottom line is, that's a mutation that led to albinism, the idea of something is no longer making any pigment. Now, the fact that it doesn't make any pigment, did it kill the organism? No, it's non-lethal. So it doesn't matter just what a mutation per se, it has to be really consequential.
Okay.
So it's all context driven in that scenario.
Just to check, but you said that neutrality is a missense mutation type?
Right, so neutral means like now when we look at this, that's what this point of this was. If you change a hydrophobic to a hydrophobic, typically that results in no effect. It's an amino acid change, right? Alanine became a valine. Fundamentally, I can look at the primary structure and I go, oh, it changed. But did it do anything? And we say neutral if it didn't really cause any functional difference. In fact, you would probably never notice that mutation ever, because unless you were looking for it, there's no functional change in your actual enzyme or whatever it is.
But you can't call a silent mutation neutral?
No, so go back to what silent's doing. That is the exact same amino acid. Why would it change anything? That's not context driven. That's just like the same exact amino acid. That is not the same thing. Okay, and here's the context I want you to remember. This is due to the degeneracy of the codon system. That's why this exists. This changed the meaning of the amino acid according to the codon list. We've now turned the page on it and go, oh, now that's coding for something different. Not the same. Does that make sense? Yeah. Then it's recycling the words. Does that make sense or nonsense? All right, nonsense. Let's talk about some nonsense. That's all I talk about over here, nonsense. All right, so now we're basically going to get to a place where this T becomes a G instead of a C. So that mutation, again, is still a point mutation. Actually, let's review. The T becomes a G. Is that a transition or a transversion? You got a little cheat sheet over there, don't you? It's transversion, right? It's a different class. So the first level is I can categorize it right there. Right, that's how I first categorize it. We have a transversion. The transversion specifically is the T becoming a G. Now we're going to ask, where did it hit? And it hit in an exon. Now we hit in an exon, so now I want to find out what happened. It's not silent anymore. It's actually nonsensical, meaning it stopped it. All right, so let's look at a protein structure theoretically over here. Just got some random globular protein right there, right? And let's say the mutation happens like right here. This is like the N terminus and the C. If you remember what that means, that's the beginning, and it winds all around to the end, right? And they're made in a series all the way to the carboxy terminus, right, the end. So if I have one that's like, say, 6 amino acids in, and it hits a stop codon now, you've truncated everything. This is all gone. This is the protein you just made. It's a peptide now. It's not going to function the same way. No, not a chance. Not even a chance it's going to. So that's going to be really bad, right? This is not like missense level. It's like you've lost the protein, period. It's like gone, right? Okay, my context driven says what happens if it's like right there. So you made everything. Let's say this is the enzyme site right here, the Pac-Man right there, right? And then you chop off that little tail, right? Who cares? Is that gone? Yeah, we still do everything we needed to. So nonsense, typically a stop codon coming up early or prematurely, you could say, before it's supposed to, is likely going to be really bad. But you could get away with it if you got lucky and it got, put at the very tail end of it, if you will, right? Everyone see that? Okay, so already we have a hierarchy. Silence, fine. Missense, we'll have to figure it out now. Nonsense, that's not so good. Now we're going to get pretty bad juju right here. This one, it's going to shift everything. All right, so frame shift means an insertion or a deletion subtype. So remember we said point mutation, you have base substitution, which is basically everything above it. And the final one means either or. Now, things are read in triplets, correct? So if I insert 3 nucleotides, or I delete 3, you're still in frame. Does that make sense? I could pull out an amino acid, and then it's just going to read the next 3 because they're in frame. Or I can delete, add or delete. I could put in 6, and then that would just be 2 additional amino acids, and then it's still going to read it correctly. So when we say the word frame shift, it means not in groups of 3. Everyone with me? Let that settle in, because now, if you do just 1 or 2 or 5, right, any of those off numbers, we now shift everything that's read from there. So we're going to do just the 1 base pair deletion, but it applies to any other scenario too. So what do we have here? We got TTT, TAC. Oh, looks like one of the T's is gone, like that one. And then AC is what's read next to it because it's got to just snap up, right? So basically, we just nixed one of those out. Could be one or the other. It's hard to tell. The bottom line is, we look up the code. That's now tyrosine. Now, on the surface, you would say, that looks like a missense. But it's not categorized as a missense because the damage keeps going on. The damage is, oh, the next 3 are read differently too. And now the next 3, and the next 3, next 3, next 3, 3, next 3. Now, eventually, what this is saying, you're going to hit a stop codon by happenstance, right? You're just going to keep reading 3s, And basically, let's imagine happens right here. So I'm going to start replicating. That part is your N, right? And I'm going to hit that spot. All right? It's a deletion or an insertion, whatever it's going to be, but it's going to be a frame shift. And now the protein is going to go sideways. That doesn't look at all like that one, because the amino acids that followed made it this shape now. And it's misshapen. And its function is not going to be able to do what it's originally intended. Now, it could have truncated as early as this, because as you read the 3s, you're just going to keep reading them according to the code. And eventually, you're just going to hit a stop sometime. Now, it could be sooner or later. It's just happenstance. But you tell me, does that look like it's going to be a good thing? Now, here's one weird thing is, still, if you hit it at the tail end, like where this had a little tail, and you have a frame shift right here, and this goes all squirrely, that's still the business end that works. So you might get away with it, but likely not. So in a hierarchy of damage and damage control, silent, you're totally good. There's not even any damage to talk about. In this sense, likely is possible. Non-sense, you're like, yeah, it's likely, not good. Frame shift, you're like, oh man, that thing's going to do some wacky stuff, right? It's now a protein could even take on a rogue function and do something, like negative, really negative. All right?
So this is happening in just the DNA, right? This isn't happening in the RNA that these mutations are happening?
Okay, so let's be really clear on this. This is where going back to your studies on Unit 1 is really important. So a heritable change has to happen in the DNA.
Mm-hmm.
So that's the first point.
Okay.
DNA transcribes into RNA, so that's a consequence of the transcription. And it's just going to carry through with whatever message is being given. So the RNA polymerase didn't make any errors. It accurately transcribed what the DNA told it to do. Now, if you change the RNA, that was RNA editing.
Okay.
That's a whole different topic. Remember I said RNA editing is not a mutation? Well, it goes totally both ways. So the idea is a mutation has to be in the DNA, and it's heritable, so that you pass it on to other descendants, and your DNA, if it's in a protein coding gene, has consequences, possibly, right? Does that make sense?
Yeah. But one more thing, since it's continuously replicating, is this also going to replicate, or is it just going to just kind of bounce itself out because the other ones are also replicating, right?
So replication, it doesn't care what's the sequence.
Okay.
So if you're replicating something and you alter what you're replicating, the machinery doesn't take any judgment. It's just going to, remember when you unzipper your 2 strands, you're going to make a complement to the other strand. You're going to make a complement, and it doesn't matter if it's a T, C, G, or A. It's just going to do its job. Like, if that's a T, then I'm going to make an A. So when you replicate, there's no consequences with mutations. You understand that?
Yeah.
Because
So
the machinery is not interpreting the. So what's happening here is an interpretation,
Okay.
right? So if there's no interpretation and you alter an STR, may I repeat? There's no consequence because there's no meaning that an STR has, right?
Okay.
But if you alter an exon, selectional pressure adds into this, which says, is that good or bad? Oh man, you stopped this thing early, and we need the full protein. So gene expression is where the rubber meets the road, if you will. That's when selection pressure happens. Selection pressure means you have to have a phenotype, right? Genotype leads to phenotype. Everybody knows that saying, right? Well, why is that? Because a genotype has to have an expression into a phenotype, and expression equals protein. Expression could also be RNA, because it expresses that too. So you could have dysfunctional RNAs, right? But we're really talking about DNA right this second. Proteins.
Okay.
Okay?
Mhm.
All right. Okay, so, sorry about that. Let me put this down. I put it on silent, but it's beeping at me. All right, so you have to commit this to memory. For sure it's on the test. Okay.
2 questions. So this is all a point mutation, right?
Yep.
And then second, the sequence for the missense mutation on the board, that would not be considered neutrality, right? Because
Well, we'd have to find out. So neutral just means the context of the actual protein, right? So this could be non-consequential. But if I look at this right now and I change the class of the amino acid from leucine, which is hydrophobic, to a polar to non-polar change, that could have some consequences. I still have to know. I'd still have to do the experiment. You'd have to say, did the enzyme change? Did the enzyme faster, slower, doesn't work? Or does it do the same thing? If it does the same thing, that equals neutral. Right? All right, just quickly over this one. This is the story I was talking about. Sickle cell anemia has one amino acid that's changed due to one nucleotide changing, which is a missense mutation. And what it does is change that glutamic acid, that's a negative charge, to a valine. So now we're actually going the opposite way of polar to non-polar. And that is actually on the surface of the molecule somewhere, exposing it. So it's an exposed hydrophobic region as it's stained. And it wants to sequester that away. It does not like the hydrophobic sitting out there. It's like, oh, this is like it's hurting it, kind of like, ah. So what it's going to do is bury that hydrophobic into another subunit that it should not be interacting with. So in other words, a tetramer, we don't have to get into too hardcore details, but I think you guys remember alpha and beta subunits, and a tetramer. So let's just say there's your tetramer comprised of your 4 subunits, right? That little just ball right there. So basically this little spot right here is hydrophobic, let's say right there. It doesn't like being there. So you guess what? This tetramer is going to bury it. Do you see how that works? It's using another subunit of red blood cell, a hemoglobin, to actually bury this into itself, and it's no longer exposed to water. This is exposed. This is not exposed. They're tucked in. So it's going to form a chain effect. So sickle cell anemia means it forms rods. And inside your red blood cell, it should be individual hemoglobin just floating around free. They should not form a chain. And if they do, that's what it's going to do. It's going to form a nice long rod, and that's what this is doing, is forming a rod and making it misshapen. All right, so that's just one nucleotide change. That's just a fundamental, like, whoa, that's got some consequence to it, right? All right, now, I often test on this concept. Even if it's not in the exon, you could still be damaging the expression of the gene. Now let's talk about what expression means. You're going to express, meaning like you make something, it expresses it, and then it has a certain primary structure and secondary, tertiary, and so that's the function part, right? So let's say that's all fine. It's all fine. But what you mutate now is like the promoter or a regulatory element. Let's deal with those first two. That means you make either too much of it or too little of it. So supersize everything. That's the way we live, right? It's always good, right? No, everything's got to be in the right amounts, right? It's like the Goldilocks principle, the idea of too much of something, too little of something, versus just right. So the just right means that there's a rhyme and reason the way the gene is supposed to work. You're supposed to have X amount of it at a certain time, and that's appropriate. And that structure needs to be as it is. So what you're changing now is not the structure. You're changing how much you make. So if you alter the promoter so that you can't even recognize it, think about that. You alter it so badly that when the RNA polymerase comes around, it's like, I don't even see a promoter. It's like, what? No promoter. How about you alter a regulatory site which makes it more active or less active? Then you make more of it, or you possibly make more of it at the wrong time. There's a whole variety of things that can happen. But the point is, the protein is the protein. Now you're altering how much you have. Does everyone see that? So you have not altered an exon. Let's go back just briefly. These are all exons. You are not hitting any of those. You are hitting outside of those areas. Right? Everybody with me? So even though you hit an exon, you still can damage those proteins of like how much you're making. Now, if you hit the 5' UTR to 3' UTR, remember the untranslated region, which is in the mRNA, by the way, right? We just tested on that. This could be in a bacteria where the ribosome binding site is. Remember how important the ribosome binding site is? It locks in on it, and if you mutate it so the ribosome cannot find it, game over. It means no protein is made. Everybody let that sink in, right? You're not going to make it if the ribosome can't find it. How about 3' UTR? Let's say you damage the poly A signal sequence. That's in the 3' area. There's a variety of places you could damage the 3'. How about the stem loop structures? Something you alter that changes a fundamental part of it. Yeah. Then the stability could be all messed up, no poly A tail, a variety of things could happen. So again, that's not an exon. So you're still messing up the protein, right? You're messing up the amounts or quantities it has. How about splice sites? The splice site is going to be inside the actual exon. It's at the edges. And so right in the intron, there's some sequences right there that are really consequential, right on the edges. And I pointed them out to you that they actually are locked in like a G and a U or something like that. They're right on the edges. Once you mutate that, you won't recognize it as a splice site anymore. And if you do that, you might have an intron that still remains in your polypeptide that's not supposed to be there. Or you can't splice it there and not change something. So that's a long way of saying, I went through the list. None of these are exons, and you can still be affecting gene expression is the way you would say it. You're affecting gene expression. All right? Everybody with me? Questions? So I asked, let's see if there's actually an eye clicker now. If I ask a question on a test that says, if you have a mutation, it's going to change gene expression, what are you going to say? Is that true or false?
True.
If you have a mutation, period, just mutation, is that going to affect gene expression?
Not in all cases.
Okay, everyone starting to clue in? Could hit an SDR, could hit junk DNA, it could hit some place that's 98% not exon, right? So the luck of the hit, wherever it hits, if it hits an exon, we have to figure that one out. But it could also hit one of these, but it also could hit something else that's totally non-consequential. I've been driving that message home, so I'm hoping you hear it.
Could you just give me another example? You might have said for
Sure.
the regulatory element.
Oh, sure. Okay, so we haven't really covered regulation, but remember we talked about, here's a gene, here's a promoter, here's a gene that you're going to make, right? Now, regulators act at a distance. So this would be like what we call an enhancer that would tell this promoter to work. So these are what we call transcription factors. We call them STFs when we talked about it before. A specific transcription factor will bind here and relay the message over here and say, turn this thing on, make it. Now what happens if you take that away? You're not making it.
And then you have inhibitors that do that too, right?
So yeah, you can do the opposite, right? The silencer version, yeah, that's just more of a, it's now not going to make it, but you can go the opposite way. You're supposed to silence, and now you make it inappropriately, right? You're making it when you shouldn't be. Any more? Sure. Okay, moving on then to the final definition. There's your neutral. Changes in amino acid sequence. That's a missense, but does not alter the function. And I bet you money that we're going to have to tell the difference between silent and neutral. They kind of sound the same. They're not, right? We already talked about it. All right, deleterious are really just the bad ones. That means that something potentially can happen that's going to be bad. Up to the worst is lethal, right? So it'll cause some problem. Now lethal, we're going to talk about that in Unit 3, Exam 3, but you can have late onset things, like the classic Huntington's disease. You still have a mutation, but you don't die until like after 50 or so, where the onset is. So lethal is relative in the sense that it could be early in a fetus, it could be as later, but that mutation plays out in a very negative way. Beneficial are far few in between. Basically tinkering with things typically doesn't end up in a great result. But this is how every once in a while you get a mutation that allows an organism to survive even better. And that's how we've changed the landscape of things. Like every once in a while you're gonna get one that all of a sudden is a paradigm shifter, and then that's actually fortuitous. Thinking about like a giraffe's neck. A giraffe's neck didn't all of a sudden just get that way, but it all of a sudden became fortuitous to have a long neck, which might be kind of freakish to some of them, like, why do you have such a long neck? And the other ones don't. And he goes, well, I can eat all this stuff that you can't, right? So that's where you might call him a freak, and then you're like, oh, well, like you can eat all the food you want, huh? All right. Now the last one is going to take a little description. Conditional. And obviously the word, follow the word. Under one condition, you're fine. Under another condition, not so fine. Up to even called conditional lethal, right? So these actually turn out to be kind of an interesting group of mutations, and it's all about what the condition is, right? So let's imagine temperature sensitivity. Now it could be heat sensitive or cold sensitive. Let's do the heat sensitive one. So that, just for the example. All right, so let's imagine this polypeptide, a globular protein, and then you mutate, let's say something right there, and for all intents and purposes at room temperature, everything's fine. It's not really a problem. So it's like goes unnoticed. Now you put a little pressure on it, right? The pressures jack up the heat a little bit, right? So now you move to like hotter temperatures, and then all of a sudden this mutation was trying to hold on, and then it's gonna denature. It's gonna unbuckle because that amino acid changed its properties such that it's not under such pressure now, but when you put it under a little bit more pressure, it's gonna break, it's gonna fold, and not do its duty, its job. This could be any condition you talk about. You change the salt concentration. You change elevation, right? So some people get really bad elevation sickness and things like that. People that have lived up in the Himalayas and different places have adapted to high elevation status through mutations that were fortuitous, right? But probably take them into a different environment might not be the same way, right? So the bottom line is a mutation under one set of conditions, no big deal. You move into a different set of conditions, whatever that is. So it just means condition, that's generic, right? All right, get it? Now, the cool thing about them is, if this was a lethal mutation, lethal gene, a gene that's really important to life, you can't study those kind of genes if it's lethal and you just kill the organism dies, right? So conditional lethals are actually been really important for science because under one set of conditions you can grow them, and then you can study what happens when you put them under this other set of conditions that cause lethality. And you say, what happens? Why is this protein so important? What is this thing? And in fact, the way we've studied genes is reverse. It's like when we find something going wrong, we go hunting for what gene that is. Notice how the names of genes are listed. Huntington's disease. There's a gene called the Huntington's gene, right? And it basically doesn't cause disease normally, but we're hot to trot on what it does after we found there's some serious dysfunction, right? So an idea is of disease and things that allow us to go trace it. But this is a cool, I know I'm not going to test on this, but per se, but the idea that you can allow it to live and then you can study it under another set of conditions is actually scientists' friend. It's been used quite a bit. All right, any questions on those? That's kind of the newer one, right? Yeah.
I was just gonna ask, for the conditional mutants, are those only identified once the conditions have
Yeah.
been checked?
So it goes unnoticed again until that situation happens, like, oh man, that's not good, right, at that moment. Yep. Okay, now we get into, it's a somewhat confusing terminology, but it's not if you kind of think about it just very rotely what it's saying. A forward mutation is classified as something that changes it from the original to the new mutant. So I'm going to use this term WT a lot, probably coming out from here, wild type. So wild type is used very much in Drosophila research. It's that kind of where it kind of established itself, and then it's kind of been co-opted. Right. It means like what's normally found. Now, does it mean it's normal? Normal's a range. Normality is not anything we can specifically say this is the prototypical genotype. Things can change, things can mutate and still be functional, right? And the idea of this, what it means is that this is the common form. So if I basically take a sampling, let's say a fruit fly, I'll use that because we're going to get to that eventually, and I look at what color is their eyes. That would be their wild type eye color, and it turns out to be red. But if you grabbed a white eye fly and you had no context, then you wouldn't know if that's normal or it's not. That's the only fly you ever looked at. But now if you look at 100 flies and 99 of them have red and this one has white, that's the outlier and that's the mutant. And the red is the one that's commonly found. So back to this idea. This gene is the wild type. It's the one that's normally functioning or common. And what's going to happen is a mutant. So a mutation. So I'm just going to call it the mutant form. So this is what we call, obviously, forward. The forward, it's not like forward thinking. It's like forward meaning it moves to that, right? It's happening like pushed in that direction. Now reversion, I'm going to use the last word because that's more the biggest important concept, means somehow it's going to get to wild type. It's going to go back. So this is a second mutation. And we call it a reversion, or you could call it a reverse. But the reason why I'm not going to lean on that word too heavily is because the context of reverse means the exact same mutation that happened here. So that would be forward mutation. Might as well just finish the diagram. Okay, the forward mutation, let's say, changes a C to a T. What kind of mutation would that be? Transition. Single ring to single ring. Just quizzing you. All right, then you take a T back to a C. Oh, voila, you're back to normal. All right, back to, okay, that's very unusual. It's very unusual that this is like lightning happening at the exact same spot twice, bam, bam. Now lightning, often a lightning storm hits in a locale, similar areas, right? But it's not going to hit always that same tree. Well, if you're carrying around a lightning rod, maybe it's going to hit you a couple times. But just the odds of things are, this is unlikely to happen, and that's like reverse, an exact reverse, like right, it goes back. Now here's the concept. Reversion is a phenotype. Now what do I mean by phenotype? When you look at the outward result, maybe it went red to white eyes, and then the second mutation brings it back to red eyes. Now, do I know how it returned back to becoming red eyes? Not necessarily. It could have been the C went to a T, and the T went back to a C, and then I got my red eyes back, right? But how about if you hit another spot, another location, and it still effectively made the red eyes, right? So imagine, okay, we're going to run in through some circumstances that are put this into play. But the idea is all you're waiting for is the actual outward phenotype. All right, let's do an enzyme then. Let's do that one instead. The enzyme doesn't work. Then you have a second mutation. Voila, the enzyme does work. Do you know what happened? Not necessarily. I know that's a reversion. And the reversion can be subclassed to several types. But I don't know that a C went to a T and a T back to a C. That's only one scenario. Everyone follow me? So reversion is about, oh, the enzyme now works. So now we have to go figure out how did that happen? So it's a second mutation called reversion, and we're going to have two classes. I don't really like the term suppressors, so I usually just say intragenic or intergenic second mutation or reversion type. Okay, so here's the deal. Suppressors have more context with like cell cycle and suppression and cancer, so they use that terminology also, so it gets a little muddled. All right, so basically here's what the deal is. Intra means what? Within. Stick with it, because that's going to help guide you. Within the same gene, you're going to have a second mutation. Intragenic, introgene. Intergenic. Give me the definition. What do you think that might mean?
Outside of.
Outside of meaning another gene, a different gene. All right, so within the same gene or in a different gene. That's all. So remember, just classifying things. So this is just categorizing. Now, there are several explanations up here, and you don't need to know them all. And certainly, I'm not quizzing you on the specific examples. But here's what I might have tune your ears towards. If the mutation happens within the same gene, intragenic, whatever the scenario. If it happens in another gene, you just classify it as inter. So let's go through intra first, because it's fairly straightforward. Notice the picture here. I like this example, because basically what it's showing is a transporter type. Like say it's an ions, and it's an ion transporter through the membrane, whatever it's going to transport. So the first mutation makes transport inhibited. That's when we were talking about like doesn't work anymore. Like an enzyme stopped doing what it's supposed to, or a transporter can't do it anymore. So it has that little lump in there, right? So that's the structural change that caused some dysfunction. Now, this one is a second mutation, the blue, right? It's not the same spot. It is not a reverse mutation, but it is a reversion because it can transport can occur again, but it happened at another site somewhere else in the same gene. Now, if you notice it, somehow, like, I would probably say kinks in a certain way. I would actually draw it a little differently. Let's say if you got this. So that would be the original one. What happens to this one then? Just buckled out that way and create a better chain. Basically, it's a compensating mutation. It compensated for it. It didn't change the original one. But when you mutate, excuse me, at the second place, you restored the function, right? So if I tilt this way, but then I tweak that, then it works again, right? First tweak, oh man, all right, I'll tweak with you. All right, works, right? So second site mutation, maybe at amino acid 15 instead of at the same place, number 6. Everyone got it? Kind of straightforward. The rest of these are kind of, some of them get a little far-fetched, but the last one. But here's one, let's see. This is the one I want to do first. This is the one I like better. This one. This one is like intragenic, except it's a subunit interaction. So I want you to see, look at 2 polypeptides. They're different colors. So we hit the first one with the first mutation, and that's called the forward. It's damaged goods. Doesn't do its job. The second mutation, wait for it, another gene, right? So it's a different gene, and they interact together. So if you hit it at the right spot, it compensates. And it's like, you hit this one, it's like, oh, I can't do my job. Well, how about if I buckle with you too? But that's 2 proteins interacting now. Let's review that, the first one. 1 polypeptide, 2 mutations within itself, compensating. And now this one is 1 mutation in 1 gene, and now the second mutation in another different gene, thus intergenic. Everyone with me? All right. Everything else has the same flavor. It's just that you have to follow some of the examples, which, let's just do a couple of them. Here's 1 gene, and here's another gene. Again, same concept of colors, right? This gene takes a hit, you lose the enzymatic function that it was supposed to have. Then you get another hit to a different gene, and it gains the enzymatic function that this one lost. Now that may sound far-fetched to you, but it happens because genes have duplicated over time and have gene family relationships, like histones. Remember I told you about how a gene duplicates and then duplicates again? So you might have a family of related enzymes, very similar. So changing its substrate of which it works might just be a matter of just tweaking it right there. And then it can do its job, right? So it becomes active again. Now, was it the same gene? No, it was a different gene. And then you compensate it back, but in a different way. Here's one more example that I'll just cover. Here's the pathway of one enzyme and a second enzyme. And it has to be, so basically at the end result, you get a certain amount of product by the speed of the enzyme working fast. And then this one's a little bit slower for it to finish the process. Well, if you get a mutation and they're both slow, then you get barely any product, right? So it's actually dysfunctional. But what happens if then you get a second tweak to this enzyme, and now it gets faster than it originally did, and that basically compensates for the whole thing. The same amount of product is basically made. So again, it turns back to wild type. And wild type is registered by the same amount of product. I don't see any difference. And where did the mutation happen? In another gene. All right? Did we beat that enough? I think you're ready for another clicker. There you go. When it says does not interact, I mean does not influence each other in either way, in any way. Does not influence each other. All right, go 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Okay, most of you got it. A lot, good majority. It's true. And why? Because this other gene has no relationship to the other one's function or whatever it's doing. It has to be related to it in some way to cause it to restore it back to wild type. So this would be actually called a first mutation for this other gene. It would be a forward mutation for that one, because it has nothing to do with the concept of what we're talking about here for going back to wild type. They're doing two separate things. Does everybody see the registry on that? We've got 22,000 genes, right? So you take another hit in another gene, but that's just another hit. The question is, did you restore back to wild type? They have to be doing something related, right? Let's see if we can apply that to the next one. Actually, technically, this should say reversion. I should change that. All right, 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. All right. Good. Even more of a shift to the, you got it. The idea is it still has to go back to wild type. It didn't revert it back, so it's just another hit to the system, right? It's still damaged goods, right? So it's got to go back to wild type is the key idea. All right, so last 5 minutes, I'm just going to start to cover this topic. By the way, this idea of reversion, don't shelve it. It's going to come right back. Later when we talk about the Ames test, you're going to be talking about reversions, and that for sure is going to be on the exam, the Ames test. So, having said that, try to understand what we just went through. Now, this right here is the idea of where the mutation hits us and then what the timing is. So, the types of mutation that hit the soma, body, somatic, right? So, somatic comes from body, soma. Somatic. So that would be your body cells, in which are the shrouds for basically our gametes, right? Some people can look at it as rotely as we are protecting the seeds, right? But in essence, that is not going to be heritable to the offspring. So it happens to us, like in our generation, I maybe had cancer or something here, but it doesn't mean our child's necessarily going to have a cancer of having that same mutation happen right there. Now they can have, we're going to change that when we talk about in lab at some point about the idea of predisposition for something means you're maybe one mutation away from causing something to happen. You might be more prone to something. That doesn't mean you're absolutely going to get it when you hand it down. All right, but that's the idea that a mutation happens somewhere in the body, and it's not going to be passed on. That happens to you locally. Now, the opposite is when it happens unknowingly to the gametes. So why is it when we get x-rays, we put protection around our genitals, our gonads in essence, right? Because we don't want the damage to hit that spot. If we're getting as much as we can, we don't want the damage to get there, because that could be, yeah, let's be clear. We're talking about a very small subset of cells in our body. We got trillions of cells, and the idea that we've got a subset that are giving rise to sperm or egg, it's a very small subset. But you hit that right spot, you hit that position, and the gametes that are your sperm-generating cells called spermatogonia or oogonia, which means they're the stem cells. Then you have the gift that keeps on giving then off to the children, and you're not really affected. It's just the sperm are affected or the eggs. So that's kind of bad one, and it goes into generations without even knowing it, right? So that's why, again, we want to protect those cells, right? So you got two categories. Ones that go germline, and those are the insidious ones. And then this is just your somatic to yourself, right? All right, the last thing I'm going to talk about is this concept right up here in the last couple minutes. So the idea of when do you get hit with a somatic mutation? So here would be the sperm and the egg unite. There's your zygote, your first cell, right? And then it goes to 2, to 4, and so forth. Trillions, right? Okay, and that's as far as I'm going to go. All right, so basically, just imagine it keeps going, right? Now, imagine if this is where the mutation happens, right there. 2 cell, then it's like this whole body is fully affected. So in early development, you get tagged then for a mutation, which is still possible to have it happen anytime, especially if you have rapid division. You get hit and a whole body plan, like half bodies, like affected. Alternatively, it could be just this one. Let's say in your 30s or somewhere, all of a sudden you have a mole, and then all of a sudden took some UV damage, and then all of a sudden it turned cancer, or turned into spreading cancer, right? But that's basically only the subset of cells that were being derived from this one. Everything else is fine. So that's kind of like the picture. That's like the little shoulder area is what's affected. All the rest of the skin is fine, right? So the point is, it's all relative to where it happens in a developmental stage and cycle about how devastating it could be. But this one is it's heritable to everything it derives all its ancestors, descendants from. Does everybody get the point? All right, so we'll come back to a figure like this too when we talk about some experiments. But that's it, and you guys have a good weekend.